Hey everybody, welcome back to the class. In this video, we will discuss Chapter 25 from the text, the urinary system. So a big part of maintaining homeostasis, that stable internal environment, is being able to maintain the composition, the pH, and the volume of our body fluids within a certain normal range, a certain limit. And a system that plays a big role in this is the urinary system. And the urinary system is responsible for removing metabolic waste and substances in excess, like excess drugs, excess toxins, from the body. And the parts of the urinary system include a pair of kidneys, a pair of ureters, the urinary bladder, and a urethra. All right, here we have the illustration of the urinary system in general. You have the kidneys here, right kidney here, left kidney here, the ureters that come off of each one, and then to the bladder down here, and then the urethra here. All right, we'll first start with the kidneys. The kidney is a bean-shaped reddish-brown structure with a fairly smooth outer surface. In a typical adult, it's about 12 centimeters long, about 6 centimeters wide, and about 3 centimeters thick. Now, each kidney is enclosed in a very strong, very fibrous capsule that helps the kidney to maintain that distinctive shape. Now, the kidneys will lie on either side of the vertebral column in the posterior abdominal wall. So this is described as being retroperitoneal. Retro meaning behind, and then the peritoneal cavity. So these are behind the parietal peritoneum. This is why if you have issues with your kidneys, like kidney stones, the initial pain almost appears as if it's a lower back pain. And even though these two organs are paired, having a left kidney and right kidney, they are not positioned in the same way. The left kidney is about 2 centimeters higher than the right kidney. And you can see that on this image again. This structure, this kidney, is higher than this one. And the reason why that is because the liver would be right above here on the right kidney. But it does not interfere with how either kidney functions. It just happens to be that one is higher than the other. All right, talking about kidney structure, the kidney has two very distinct and very different regions, and these are called the cortex and the medulla. And these are terms that are very common in anatomy, in biology in general, so don't associate cortex and medulla with just the kidneys. Those have a particular meaning. The cortex of a structure is going to be the outer part of the structure, and the medulla will be toward the middle. So we think of M for middle, M for medulla. So the renal cortex would be the outer part of the kidney, and then the renal medulla would be the inner part. And of course the word renal is always referenced to the kidney in general. Now there are portions of the cortex that actually will project inward toward the center of the kidney. And these projections are called renal columns. And they're called that because they look like the columns that you would see on a building, for example. And these columns will form sections within the medulla called renal pyramids because they have a pyramid-like shape to them. And there's a structure that you'll find on the tips of these pyramids called a calyx. So a calyx is a singular term. When you talk about these in plural, that would be called calyces. All right, so here we have an illustration on the right and a real kidney on the left. So the cortex, the outermost part here, you can see is a lighter color. The medulla, here you can see it's a little bit darker in its coloring. And I'll point out the columns and the pyramids. On the illustration, you can see how the cortex here dips down here, and here, and here, 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 and here. Those are inward projections of the cortex. So even though they're going into the medulla, the renal columns are a part of the cortex. And you can see that here, there. So it's a very easy way to distinguish the different coloring, which indicates the cortex versus the medulla. And these pyramid-shaped structures are the renal pyramids. And you can clearly see those here because they have a much different coloring. And at the tip of each of these pyramids is where you will find a calyx or calyces. So the columns are part of the cortex, the pyramids are part of the medulla. Now when it comes to the calyces, there are different kinds. You have some that are smaller and then some that are larger. The smaller ones are called minor calyces. And once you have several small ones of those merging together, that's what will form a major calyx or major calyces. The major calyces will merge together to form what's called a renal pelvis. This pelvis is a funnel-shaped cavity that collects all of the urine that is drained from all the pyramids. And the pelvis also forms the most superior end of the ureter. So going back to a different image here, the renal pyramid here, here, here. See, at the tips of each one of these pyramids would be a calyx, a minor calyx. Then when you have several of those smaller minor calyces, will form a major calyx. When you have several of these major calyces formed together, you have this entire basin-like structure here. That's the renal pelvis. So that's where all of the urine that gets collected by this pyramid and that pyramid and so on all gets dumped and collected into the renal pelvis. And that renal pelvis is the most superior end of the ureter. This is the tube that leaves each kidney that goes on into the bladder. Alright, next is a structure we've already talked about before in previous chapters, and that's the term hilum. Just like we saw with the lungs, any indented area where you have larger vessels, nerves, lymphatic vessels entering a structure is going to be called the hilum. Well, you have the same setup here with the kidneys. So the area where you have larger vessels, the ureter, lymphatic vessels entering the kidney is the renal hilum. And the hilum will lead to a much larger hollow chamber that's called the renal sinus. And the sinus is a cavity within the kidney that contains everything: the renal pelvis, the renal calyces, blood vessels, nerves, fat, everything. Alright, next we'll talk about some important blood vessels associated with the kidney. Of course, the main one would be the renal artery. This will enter each kidney through the hilum, and it will continue to branch further on within the kidney to form the functional units of each kidney called the nephron. And the final branches of the renal artery that will actually enter each nephron are called the afferent arterioles. Remember, any structure that is afferent or afferent is something that is arriving at a structure. So there's going to be an afferent arteriole and an efferent arteriole. Those would end up leaving the nephron. Alright, here is a model of a typical kidney with all of the nephrons that have been dyed. This to show you how complex it is. And you can see the parts that are in red would be branches of the renal artery, which would be right here. Those are going into each individual nephron. Then the blue would be the venous system going back to the renal vein, which would be this structure here in blue. Now, each kidney contains about 1 million nephrons. And again, that's an approximation. It'd be difficult to get an exact number. But there should be right around 1 million in each kidney. The nephron has two main parts. You have the renal tubule and the renal corpuscle. The corpuscle is a filtering unit that is composed of a tangled clump of capillaries called the glomerulus. And that mass of capillaries is surrounded by a sac-like structure called the Bowman's capsule. So the corpuscle is both of those together. So the glomerulus is one part of it, Bowman's capsule is another part, but the term renal corpuscle refers to both of them together. Alright, once the blood enters the glomerulus through the afferent arterioles, blood will get filtered through that mass of capillaries. Then the blood will exit the glomerulus through the efferent arteriole. Remember, afferent is arriving at a structure, efferent is exiting a structure. And this filtration in these capillaries in the glomerulus is the very first step in the forming of urine. Here is a close-up view of how this would look. This tangled mass, all of this, that is the glomerulus. And then the area right around it is the Bowman's capsule, sometimes called the glomerular capsule. Either term is fine. Then the tubules will be found just outside of that. This is how this area would look in a histological stain. You can clearly see the tangled masses here and here and here. That would be a glomerulus here. The area right around it would be the Bowman's capsule or the glomerular capsule. And those two together are referenced as the renal corpuscle. Now when it comes to the nephron, the structures that enter the nephron that are around it, it's important to understand what these structures do. And the names can get a little cumbersome, but the name is telling you exactly where it is and exactly what it's doing. All right, so the renal tubule will lead away from the Bowman's capsule. And this first part of the tubule is very, very coiled. This initial section of the renal tubule has a very specific name. It is called the proximal convoluted tubule. It's called that because it's proximal, or very close to the glomerulus. It's convoluted because convoluted means very coiled or very twisted. So these terms, proximal or distal, in this aspect are referencing where the tubule is in relation to the glomerulus. So the tubule will then continue on into the medulla of the kidney, and it will form what's called the loop of Henle, or the nephron loop, as some people reference it. And it has two different parts. You have a part that goes down and the part that comes back up. So the part that goes down is the descending limb, and the part that comes back up is the ascending limb. And from here, the tubule will continue on, moving further away from Bowman's capsule, and that part is called the distal convoluted tubule. It's called distal because it's further away from the glomerulus, but it's still coiled and twisted. The distal convoluted tubules, several nephrons will empty together into a collecting duct, and then these ducts will continue through the medulla to join with other collecting ducts to drain into a minor calyx. All right, so again, a typical illustration of a kidney that we've seen multiple times already. We're going to zoom in on this one area here, this rectangle. That's what this is. And then from here, we're going to zoom in on just one nephron. So you see a bunch of those here, but we'll zoom in just on one. And this is the image that you should be familiar with because it's very likely to come up again in the future. And the key to this image is following the arrows, because the arrows are telling you in what direction things are flowing. The arteries here, blood flow is going this way. Blood will enter the glomerulus, which is this tangled mass here. The area around it would be Bowman's capsule. So this structure here, how the blood enters this glomerulus, is the afferent arteriole. Blood will get filtered in here. When blood leaves this glomerulus, it will leave through this vessel, the efferent arteriole. Then it will continue on through the venous system and then back on into the heart. The material that is filtered by the glomerulus is caught by the Bowman's capsule. That's why this is surrounding this mass of capillaries. So it's there to catch whatever gets filtered. This first part of the tubule, the proximal convoluted tubule, because it's close to this structure and it's quite coiled and twisted. At this point, it will go down into the medulla. That's what this dotted line is indicating. Anything here down is the medulla. Anything above that line is the cortex. So you have the nephron loop. You have this part going down and this part going up. So that part is the descending limb. And this part is the ascending limb. But together, that is the loop of Henle, or the nephron loop. Then that tubule will continue on. It gets a little bit more coiled over here. This part is the distal convoluted tubule, because it's a little bit further away to here, but it's still quite coiled and twisted. And that will lead on to a collecting duct. And from there, it will meet up with other collecting ducts to form a minor calyx. So this is just one nephron out of 1 million found in each kidney. So you're going to have around 2 million of these. This is how urine starts to get formed. Now there are two different kinds of nephrons, and they differ in two important things: in how long the loop of Henle is, and how close their capsule is to the medulla. So the two kinds are cortical or juxtamedullary nephrons. A cortical nephron will be very far from the medulla, and these will also have shorter loops of Henle's. Of course, the opposite is going to be true for the other kind. For the juxtamedullary nephrons, these are going to be very close to the medulla and have longer loops of Henle. Right, so for here, we are comparing this nephron here versus this nephron here. This one, you can see the Bowman's capsule is further away from the medulla down here, and they have shorter loops of Henle. That's going to be a cortical nephron because it's more in the cortex than it is in the medulla. For this nephron, this Bowman's capsule is quite close to the medulla, so that is juxtamedullary because it is right next to the medulla portion of the kidney, and a much longer loop of Henle compared to this kind. So know how those two are different and be able to identify either one either by regular text question or with an image. All right, next topic we'll talk about is the formation of urine. The main function of the nephrons that we just talked about is to control the composition of the body fluids and to remove waste from the blood. And by doing that, you are forming urine. And urine is made up of electrolytes and waste and, of course, excess water. And the formation of urine is due to three distinct processes. And we've already talked about the first one, glomerular filtration, where things get filtered by the glomerulus. Whatever is filtered is caught by the Bowman's capsule and then put into the proximal convoluted tubule and so on. So that part we've already talked about. Now the next two processes are very, very easy to get backwards. And because the names that go along with each one seem counterintuitive. And that's tubular reabsorption and tubular secretion. I will talk briefly about glomerular filtration because this is something that we've already discussed when we talked about the nephron and the glomerulus. So smaller items like ions, some small dissolved particles, water, can be filtered by the glomerulus, but things that are larger can't because of their size. The capillaries that are found in the glomerulus are a lot more permeable than other capillaries that we've talked about. These are going to be fenestrated capillaries. And anything that is filtered and then caught by the Bowman's capsule, at that point is then called filtrate. This image, we're going to zoom in just on the glomerulus, just on the Bowman's capsule. So you have the blood entering here through the afferent arteriole. Blood will get filtered here. And as the blood leaves, it will leave through the efferent arteriole here. This tangled mass is the glomerulus. The cap around it is the Bowman's capsule. So whatever gets filtered is caught by this structure. At that point, it's called filtrate. Then it goes on. This is the first part of the proximal convoluted tubule right up here. If you zoom in just on one small piece of the glomerulus, you'll have this. And then again, a term we've had before when we talked about capillaries. Much larger holes are the fenestrae, because this is a fenestrated type of capillary. So those larger holes allow for more things to be filtered and then caught by the Bowman's capsule. Now the main force that moves substances through the glomerulus is hydrostatic pressure of the blood. And that's because the efferent and afferent arterioles have a different diameter from one another. The efferent arteriole actually is smaller in diameter than the afferent arteriole. And that difference in size causes the blood to move faster as it exits. An example that I give to kind of demonstrate that is putting your thumb over running water in a garden hose. When you put your thumb over the end of that hose, the water will speed up because the space that it has to travel through is much smaller. That lowering of the space means the velocity of the fluid has to increase. That's what's going on with the afferent and efferent arterioles. So here's what I mean by that. And the afferent arteriole here, efferent here. As the blood is leaving, it has to go through a smaller space. So to compensate for that, the material will go faster through the glomerulus. And the last main concept that we'll talk about with the glomerular filtration is the GFR, the glomerular filtration rate. And this is directly proportional to the filtration pressure. So if there's a higher pressure pushing the blood to be filtered, then the GFR will also go up. If the filtration pressure goes down, the filtration rate will go down. And things that can affect the GFR are having a enlarged prostate in men, having kidney stones, or any kind of obstruction. That's going to indicate glomeruli are not filtering as much as they should, or at the rate that they should, which could indicate a beginning sign of renal failure. All right, now we'll move on to tubular reabsorption. Substances will move from the renal tubules into the blood capillaries that surround it, and those capillaries are called peritubular capillaries. Most tubular reabsorption, roughly 70% or so, will occur in the proximal convoluted tubule, and the structure is lined with a series of microvilli. So most of the reabsorption happens right after it leaves the glomerulus in that first part of the tubule, the proximal convoluted tubule. We'll take a look at one glomerulus, one capsule here, afferent arteriole here, blood gets filtered. What can't get filtered, or what remains in the blood, will exit through the efferent arteriole here. Of course, you have Bowman's capsule here. You have filtrate entering here. This first part is the proximal convoluted tubule. And here is why the names of these last two processes in the formation of urine tend to confuse almost every student, at least in the beginning, because the names sound counterintuitive. Tubular reabsorption. This is where you have material being reabsorbed from the renal tubule into the blood capillaries, not the other way around. So that you would think the name indicates that there's reabsorption happening by the tubule, but it's the exact opposite of what you would think it is. So tubular reabsorption, material is being absorbed from the tubule into the blood capillaries. Now this reabsorption of water will occur passively by osmosis. Sodium and other ions are going to be reabsorbed due to active transport. We'll talk about that in a moment. And even small changes in tubular reabsorption of sodium or water can have huge changes in the amount of urine that gets excreted by the body. All right, so we've talked about filtration in the glomerulus. We talked about tubular reabsorption. The last process in the formation of urine is tubular secretion. Now this is where you have substances moving from the plasma of the blood capillaries, the peritubular capillaries, going into the renal tubules. So material is being secreted into the renal tubule. So don't confuse tubular reabsorption and tubular secretion. Now this will happen when the body does not need or doesn't want certain materials, such as excess water or drugs or waste. And the secretion of hydrogen ions is an important method to regulating the pH of body fluids. All right, so we're zooming in on a small piece of a blood capillary, a peritubular capillary. This image shows both tubular reabsorption and tubular secretion. So this yellow structure here would be the tubule. When a material is moving from the renal tubule into the blood capillary, that is tubular reabsorption. It's being reabsorbed from the renal tubule. When you have material moving from the capillary into the tubule, that is tubular secretion. Material is being secreted into the renal tubule. All right, next we'll talk about urine concentrations. The ability of the kidneys to maintain that stable internal environment, that homeostasis, rests on the ability to concentrate urine by reabsorbing large volumes of water. And the different parts of the nephron absorb different things. So you need to be able to match what is going on at each section of the tubule. The first part we'll talk about is the descending limb of that loop of Finley. This is permeable to water, but nothing else. So water will leave this portion by osmosis, and will enter the interstitial fluid. When you have a whole bunch of water leaving a solution like this, what you're leaving behind is a highly concentrated solute. This is the descending limb of the loop of Henle. So what you have here is everything that was filtered and caught by the Bowman's capsule here will travel on here, but then you're going to take out almost all of the water. When you get to this point, there's hardly any water left, but you're going to have all of the solutes, all of the solid parts that are still there. That's why it's a very highly concentrated solution. Now the ascending limb is not permeable to water, and ions have to be pumped out by active transport. So this will leave a very weakly concentrated solute. So going back quickly to this image, by the time we get to here, we have hardly any water and a highly concentrated material here. As it travels up the ascending limb, ions get pumped out of the ascending limb through active transport. That will leave a very weakly concentrated material when we get to this point. Now the difference in how fluid moves between the descending and ascending limbs is known as the countercurrent mechanism, because what happens on one limb of the loop of Henle is going to impact what happens on the other. You have material moving in two different directions within the same structure. So for here, we're zooming in on one loop of Henle with some actual measurements to go with it. So let's say the concentration of this filtrate is 300. Again, through the descending limb, you get water being pumped out. So you're left with a very highly concentrated material. So we went from 300 up here at the start of the process. We're now up to 1200 because you've removed almost all of the water at this point. That's why it is so highly concentrated. As the material goes up the ascending limb, ions like sodium ion, chloride ion are pumped out through active transport, and they get pumped out so much that you have a very weakly concentrated product at this point. So we started at 300, spiked up to 1200 down here, but you ended up with a value of only 100. So you end up the process with a lower concentration than you started with. And the rate of how ions get pumped out through here on this side is going to impact what happens on this side. That's why this is called a counter-current mechanism. What happens on this side is going to affect here. On this side you have material moving down. On this side you have material moving up. So counter-current, two opposite directions of flowing material. Alright, next part will be the DCT, the distal convoluted tubule. This is controlled by hormonal actions like anti-diuretic hormone like aldosterone. Water and electrolytes are going to be reabsorbed by the tubule here. That will take you to the collecting ducts. These are also under hormonal control. And here you have active transport will help pump out any material that the body wants, and then leaving behind what it doesn't want. So at this point, it will continue on as urine because the body can't use it or it doesn't want it. And at this point, the goal is just to eliminate it. All right, some components of urine, if you know what the products are representing. We have urea and uric acid. Urea is the byproduct of the breakdown of proteins. So the amount of urea within someone's urine is a reflection of how much protein they have in their diet. Uric acid is a byproduct of nucleic acid breakdown, such as the adenines and guanines, more specifically. Probably the easiest way to keep these two straight. Uric acid, acid is in that name. It's like nucleic acid has that word in its name. All right, the composition of urine, as you would imagine, is almost all water. It's about 95% water. Will usually contain uric acid, urea, creatinine, may contain trace amounts of amino acids and some varying levels of electrolytes. The volume of the urine will vary with the amount of fluid that's taken in someone's diet, and also environmental factors. All right, after all this urine gets collected and then taken to the renal pelvis, it will travel from there through ureters onto the bladder. The ureters are about 25 centimeters long, and they will join the urinary bladder within the pelvic cavity. These run parallel to the vertebral column. The innermost layer of the ureters, also known as the mucus coat, this is where you find the transitional epithelial cells. In last semester, we talked about how transitional epithelium is only found within the urinary tract. Well, that's where these are found in this structure. They're found in the innermost layer. This is what will allow the ureter to expand as urine moves by it, but then recoil back and not lose any strength or any integrity of the tissue. So if you were to cut a ureter in cross section, this is what you would see. This inner lining here that's a little bit more purple, that is the mucus coat. So that lumen, that space that's in white here, that's where urine would travel from the kidney through this tube onto the bladder. So that takes us to the urinary bladder. This is a hollow, distendable muscular organ that we have in the pelvic cavity. There you will find smooth muscle fibers that will make up the detrusor muscle, which is the muscle of the bladder wall. You also find the internal urethral sphincter. So even though it has urethra in the name, this structure is found within the bladder. This is the sphincter that surrounds the neck of the bladder that prevents the bladder from emptying. This is why whenever you have to urinate really badly, this is why it will start to hurt. There's actual physical pain there because that sphincter is being stretched more and more than it should. All right, here we have a posterior and anterior view of a bladder of a male. And you can tell it's a male because of the prostate gland that's here. So you have the detrusor muscle here. You can see that in the anterior view. And then the internal urethral sphincter is right there. So as this bladder fills with urine, the sphincter will get more and more pressure put onto it. And then here is the posterior view. And these are structures we'll talk about in two more chapters when we get to the reproductive system, the seminal vesicles and the prostate gland. All right, from there, the urine will exit the body through the urethra. So don't confuse the ureter with urethra. Ureters go from the kidneys to the bladder, and urethra is what you pee out of. So this is the tube that will convey urine from the bladder to the outside of the body. There is quite a difference in length of the urethra within males and females. In females, it's about 4 centimeters long, but in males, it's about 5 times that length. It's about 20 centimeters long. And then also in males, the urethra has a dual function, not only for conveying urine, but also has a role with reproduction. All right, for here we have a comparison of male versus female bladder and urethra. So the image on top would be female, image on the bottom would be male. The bladder is roughly the same. Urethra, only about 4 centimeters long for females, but about 20 for males. All right, that brings us to the end of this chapter on the urinary system. Like always, if you have questions, feel free to post them on the class discussion board on Blackboard. Reminder, I do have those online office hours that are live every Monday and Wednesday morning, 9:30 to 11, also available through Blackboard. Or you can also email me or text me any questions you may have. Thank you for watching, and we'll see you in the next video.